Welcome to our lesson on osmosis. The key questions for today are: what is osmosis? How can osmosis be investigated? What happens to different types of cells when placed in solutions? How do you calculate the rate of osmosis? How is water uptake converted into percentage change in mass, and why? Keywords that you need to know are dilute, concentrated, solution, solute, isotonic. Osmosis is a specific type of diffusion that applies to the movement of water. This movement occurs across a semi-permeable membrane. During osmosis, there is a net movement of water from a dilute solution to a more concentrated solution. Where there is lots of water or little solute, that's dissolved substances, to where there is little water or more solute. Task: match up the key terms to their definitions. Osmosis is responsible for several effects on cells when they are placed in solutions of different concentrations. Solutions can be described as hypertonic, hypotonic, and isotonic. Hypertonic means more concentrated in comparison with the cell. Hypotonic means less concentrated than the cell. And isotonic means the same concentration as the cell. If a cell is placed in a solution that has a higher concentration of solutes than the inside of the cell, so hypertonic, water will move out of the cell and into the solution, leaving the cell shriveled or plasmolysed. If a cell is placed in a solution that has a lower concentration of solutes than inside the cell, so hypotonic, then water will move into the cell, making it swell up. If a cell is placed in a solution that has an equal concentration of solutes to inside the cell, so isotonic, then there will be no net movement of water. This is why cells, and therefore tissues, change in mass when they are placed in different solutions. Task: suggest what could happen to a red blood cell placed into distilled water, and how would this differ to a plant cell? The red blood cell would swell up and eventually burst as water would move into the cell by osmosis. If a plant cell was placed in distilled water, it would swell up but would not burst, as the cell wall would hold the cell together and withstand the pressure from the water. Osmosis in plant cells may be investigated using pieces of potato, cucumber, carrot, or any other vegetable that can hold a fixed shape. Water will move into or out of the cells depending on the concentrations of the solutions that it is placed in. During this experiment, the vegetable pieces are cut to similar volumes and weighed before and after being left in a solution for several hours. The change in mass can be calculated, and if the time is monitored, a rate of water uptake. For example, a piece of potato gains 0.012 grams of mass in 8 hours. The rate of water uptake would be 0.012 divided by 8, which equals 0.0015 grams per hour. Extension: why must raw vegetables be used for osmosis investigations? The vegetables used in the investigations must be raw, as high temperatures involved in cooking destroys the partially permeable membrane, meaning osmosis will not occur. Task: complete this AQA exam question. The lugworm lives in a U-shaped burrow in the sand on the seashore. The diagram below shows a lugworm in its burrow. A. Some scientists investigated the effect of different salt concentrations on lugworms. The scientists collected 50 lugworms from the seashore, separated them into 5 groups of 10 lugworms, weighed each group of 10 lugworms, placed each group into a different concentration of salt solution and left them for 8 hours. Took each lugworm out of the solution and placed it on blotting paper for 30 seconds. Reweighed each group of 10 lugworms. One, why did the scientists use groups of 10 lugworms and not just one lugworm at each concentration? 2. Suggest why the scientists place each lugworm on blotting paper for 30 seconds before they reweighed the group of lugworms. 3. How might the method of blotting have caused errors in the results? 4. Suggest one improvement the scientists could make to their investigation. B. The table shows the scientists' results. One, the scientists calculated the percentage change in mass at each salt concentration. Why is the percentage change in mass more useful than just the change in mass in grams? Use the information from the table in your answer. 2. Calculate the percentage change in mass for the 10 lugworms in the salt solution with a concentration of 2.0 arbitrary units. The following graph shows how the percentage change in mass of a potato disc changes as the concentration of the salt solution it is placed in increases. We can see that as the salt solution concentration increases, the percentage change in mass decreases, crosses the x-axis into the negative area of the graph. This shows that the potato gained the most mass when the salt solution was at its most dilute, gaining less and less mass as the solution got more concentrated. As the water was moving from the solution into the potato, then the potato started to lose mass. Task: why did the potato lose mass? The potato lost mass because water moved from inside the potato to the solution. This is because the solution became more concentrated or hypertonic compared to the potato. Extension. What can you tell about the solution when no mass change was measured? The solution was the same concentration as the potato. It was isotonic, so there was no net movement of water. When the substance is the same concentration as the potato, there will be no net movement of water, as there is no concentration gradient.